Okay, so thanks Federica for the introduction. I am Barbara Rossi from Elettra Sincrotrone Trieste, and we participate to the PRP project with our beamline IUVS that is mainly dedicated to the inelastic ultraviolet scattering. And today in this presentation, I would like to give you an overview on the technique of UV Raman spectroscopy, and in particular on the opportunity offered by this technique in the investigation of biological system. So for the people that are not so familiar with Raman spectroscopy, this technique takes the name by Sir Raman, Nobel Prize, who firstly discovered this effect. This is a relatively simple effect that we observe when a monochromatic light interacts with a sample. A part of this light is scattered. Sorry, there is a pointer. No, okay, no problem. Ce la farò mai. Ok. A metà, okay. So thanks. So when the light interacts with the sample, and the part of this light is inelastically scattered, so this means that the energy of this light changes, and the shift in the photon energy is related to the excitation or the activation of molecular vibration. So with this type of experiment, we obtain a Raman spectrum that consists in several bands, where each of these bands can be associated to vibration, specific vibration of chemical group that you have in your system. So the the Raman spectrum, that is a molecular vibrational spectra, similar to the infrared spectra, but the technique are complementary. And so the information that we can extract from these spectra are information mainly related to the molecular structure, but not only, in the sense that Raman spectroscopy gives the opportunity to provide a non-invasive way to characterize both the structure and dynamics of molecular system. The advantage of this technique with respect to the other vibrational techniques, such as infrared spectroscopy, is the fact that the technique is non-contacting and non-destructive, in the sense that the process is not a absorption process, but the light is scattered by the sample. So this means that the technique does not require sample handling or pretreatment. It's not necessary to have a thin layer of your sample, for example, similar to the case of absorption spectroscopy. And the other important thing is that water is practically transparent in the fingerprint region, where we observe the mainly vibrational band of biological samples. So this means that you can work with your sample in hydrated state, also in relative highly diluted state, without the problem of the interference given by the solvent. The disadvantage of the technique of Raman spectroscopy is the fact that this effect is a relatively weak effect with respect to the absorption spectroscopy, for example. And second problem that is related to this first problem is the fact that for this, the fluorescence emission of your sample tend to interfere or can interfere with your Raman collecting, with the collecting of the Raman spectra. And sometimes this problem of fluorescence emission can strongly interfere with the collection of the measurement, also with the analysis of your sample. All these problems can be solved by using or by exploiting the UV resonance effect. It's a very complex effect from the theoretical point of view, but from the experimental point of view is relatively simple, in the sense that when we use an excitation wavelength for your Raman experiment with an energy with an energy that corresponds or is close to the energy of any electronic transition characteristic of your sample, we observe a strongly enhancement of the Raman signal intensity that is selective only on the vibrational mode associated to the chromophore in resonance with your excitation wavelength. So this means that with the UV Resonance Raman spectroscopy, we obtain a strong increment of the detection limit of the technique. So this means that the sensitivity of UVRR technique is strongly enhanced with respect to the visible standard spectroscopy, but most importantly, this technique gives you the possibility of very strong selectivity on specific chromophoric fragment present in your sample. And so the technique is a labor-free technique, but this is very sensitive to specific molecular site of the sample under investigation. Here we can see a typical example of the use of UVRR spectroscopy for the study of protein. Here there is the absorption spectrum of protein, okay? And if you use as excitation wavelength to collect the Raman spectra, we can see that, for example, if you use an excitation wavelength at 229 that is close to the resonance energy, to the electron transition of the aromatic amino acid present in the sample, the spectra, the Raman spectra is completely dominated by the signal assigned to the tryptophan and tyrosine, phenylalanine, and other aromatic amino acid present in the sample. If you use another excitation wavelength close to the electronic transition of the amide group, the spectrum changes because it's dominated. In this case, we are able to strongly enhance the signal associated to the amide group present in the protein. Of course, this technique requires a very tunable light source, especially in the deep UV range. And for this reason, synchrotron radiation is the optimal solution. Synchrotron radiation source use the emission of relativistic electron bunch that are accelerated in this so-called storage ring. And during this acceleration, this electron produce electromagnetic energy under the form of light. And the characteristic of this light that is so-called color light, in the sense that the spectrum of the emission of the synchrotron source is very wide, typically come from infrared to hard X-ray energy. And completely, we have a completely full tunability of the spectrum of the emission in this range. Here we can see the characteristic of the Elettra storage ring that is in operation from about 30 years. The energy produced by the storage ring of Elettra comes from the energy of the infrared until the soft X-ray. And you use in particular the emission of the spectrum of the synchrotron light of Elettra in the UV range for UV Resonance Raman experiment that exploit this full tunability of the excitation source. So this fine tunability of the excitation wavelength allow to disentangle the Raman signal from selected molecular portion. Here we can see that the UVRR technique is a very sensitive molecular probe for biological sample, because in the deep UV range, so between about 200 and 300 nanometer, we can find the electronic transition of several characteristic functional group of biological sample, amide bonds, for example, in the case of polypeptide, aromatic amino acid, in the case of the proteins, several biomarkers, in the case of complex, more complex systems such as virus, or the electronic transition typical of the nucleobases in the case of the nucleic acid. The technique is an ideal tool for the study of highly diluted sample in real condition, with the possibility to make operand and in situ experiment, thanks to this fact that the sample does not require special pretreatment. Here we can see the layout of the IUVS beamline, that is the beamline dedicated to the UV Resonance Raman spectroscopy, multi-wavelength UVRR experiment. We use the emission of the synchrotron light in the deep UV range. We have to access to about to this range between about 200 until 270, 80 nanometer. This light is further monochromatized and use similar to the laser source to make this UVRR experiment in macro configuration. So this the setup at the moment is ideal for the study of liquids, solution, and gel, because we work mainly in macro configuration, and we have also the possibility to use fixed energy laser at 3 excitation wavelength, 213, 224, and 266 nanometer for special type of experiment that's not required the full tunability of the synchrotron source. This is the layout of the beamline at the present, and this is the near future, the new source of the USA 2.0 beamline. That is this upgrade is in the framework of the general upgrade of Elettra Sincrotrone, the project of the Elettra 2.0 that in which we plan a strong upgrade of all the synchrotron Elettra storage ring. In particular for our beamline, we are installing at the moment and commissioning this new source that is a laser source composed by different type of laser and harmonic generation and mixing units. The results of the emission of this laser is a very wide extension range in the deep UV range, starting from about 191 nanometer until the far UV, and also in this other region close to the visible range. So this is the scheme of the layout of the new beamline that we use this laser source with the main advantage to the very high flux milliwatt with respect to the microwatt that we use now with the synchrotron light. So this means about 100 times intensity of the light that we use for our experiment, and extended UV range, and a very fine spectral line width, so a very good resolution for the spectra. So this is the at the moment the laser is in the phase of the installation, and we plan to have active this new setup at the beginning of next year. Together with this implementation, we have the implementation of the beamline planned in the framework of the PRP project, in particular the work package 5 and 7. And here we can see the layout of the beamline at the end of this, we hope at the end of the project. Of course, the setup mainly dedicated to the multi-wavelength macro UVRR experiment in this UV extender range. The implementation that we plan, we are buying this microscope that will be coupled with the spectrometer, and so we have the possibility to collect also micro UVRR spectra. Mainly, this station will be mainly dedicated to the cytology and histology, because we add to the spectroscopic information also the lateral resolution, so the possibility to have to collect mapping and to collect different Raman UV resonance Raman spectra from different points of the sample. And with this setup will be equipped with these 3 fixed energy laser. And finally, we have this other new instrument that will be a portable, fully integrated deep UV Raman instrument for macro and micro measurement, with an excitation source at about 250 nanometer. The characteristic of this instrument that is it is possible to measure in situ, real time, and in daily light measurement. So this means that you can move this instrument for measurement directly in situ, for example, on the surface, in the detection of pathogens, or for other type of similar experiment. So now I would like to show some application of the technique of the UVRR spectroscopy, especially for the analysis of biological sample. This is the example of the application of the UVRR spectroscopy for the selective probing of nucleic acid and, in general, DNA assembly. In this case, the technique give the possibility of selective probing of the structure and environment of the different nucleobases. In fact, you see that if you consider the 4 nucleobases that compose the DNA molecule, and we use as excitation wavelength light at the energy corresponding to the characteristic electronic transition of the 4 nucleobases that you see are slightly different. In the spectra of the DNA, this is the case of the salmon DNA as model system, we can observe the typical fissure associated to the specific nucleobases. So this means that we can isolate in the spectra in very selective way the contribution coming from different parts of the same DNA molecule. And this can be very useful for different type of study in the case of the DNA, nucleic acid, but also in the more complex structure, as for example, the G quadruplex structure. This is an example of application of this technique. G quadruplex is a guanine-rich sequence of the DNA that are particularly interesting for the application in the field of the drug delivery, because this part of the DNA are this motif tend to interact directly with the drug inside. In the case, for example, of when there is an interaction with drug or other molecule. And in this case, we use for this experiment an excitation wavelength in the close resonance with the guanine nucleobases. And so we are able to enhance in the spectra the contribution, the vibrational bands mainly associated to the guanine nucleobases. And in particular, the intensity of this band, in this case, we study the intensity as a function of temperature, are strongly related to the base unstacking localized on this guanine basis. So this means that we can associate the change of the intensity of this band with the change of the base unstacking that is in turn related to the stability or instability of different DNA or G quadruplex structure. These spectra have been collected at relatively low concentration for this type of Raman experiment, concentration that are comparable to the same concentration that we use for absorption or circular dichroism experiment, and the contribution of the solvent, aqueous buffer in this case, can be completely neglected because it's not this contribution is not present in this fingerprint region. Here we have studied, for example, the stability of the G quadruplex in the presence of this drug, actinomycin. In this case, the intensity of this band follow as a function of temperature is representative of this unstacking interaction that change as a function of temperature, but change in a different way in the presence of the drug. Then in particular, we have demonstrated that in the complexation with actinomycin, there is a persistent stacking of the guanine bases that tend to stabilize the structure of the DNA thanks to the presence of the drug. This is another possible application of the interaction between the SARS-CoV G quadruplex RNA and drugs. Here we use the UV Raman spectroscopy in the framework of general project, where the main focus is the identification of therapeutic molecule able to bind with this RNA structure to stabilize the complex. And in this case, we can see that, for example, we can extract particular information from the so-called difference spectra, that are the spectra where we can subtract the contribution of the single nucleobases, and we can identify in this way the specific effect of the drug at specific site of the RNA in this case. Another application of the technique this study is a very long project in our laboratory that concerned the investigation of the stability of the DNA in so-called neotoric solvents. So the idea is to find new solvent or co-solvent able to stabilize the DNA that is subjected to chemical degradation when it's stored in water or aqueous buffer for a long time. In this case, we use mainly two different excitation wavelengths, 250 and 266, to enhance in the spectra the specific band related to the adenine and guanine nucleobases. And from the intensity and the frequency position of this band, we can extract information of the hydrogen bond breaking and base stacking that involve these nucleobases, but also information on the alteration of hydrogen bonding site. This is really important in the case of the solvation of a system. Here we study, for example, the effect of the imidazolium-based ionic liquid and water mixture. When we prepare this mix and we solve DNA in this solution, we can obtain an stabilization of the DNA that can be marked by this temperature-dependent experiment where we follow the evolution of specific band, and we can in particular identify some characteristic melting temperature that consists in this strong increment of the intensity that is related to this unstacking, so destabilization of the DNA structure. And we identify, for example, in this case, different characteristics of this cation of this ionic liquid that are able to give an increment in the melting temperature. This type of experiment can be associated also to molecular dynamic simulation that can give detailed information on the type of the local interaction that are at the basis of this thermal protective effect. For example, in this case, we identify this strong cation-mediated interaction with the DNA that seems to be responsible of this more effective stacking between guanine bases in the presence of ionic liquid. Here we can investigate or we can extract other local information on the hydrogen bond, for example, on the different effect of the solvation on local site of the DNA by looking to the frequency position of some specific band associated to the guanine, adenine, or thymine nucleobases, for example. And also in this case, it's fundamental, it's a fundamental importance, the possibility to change the excitation wavelength in order to identify specific spectroscopic marker in the spectra. And another possible application of this technique is for the study, for example, of toxic effect of phthalic acid on the double structure strand of the DNA. In this case, this study was performed with a very relatively short sequence of the DNA that mimic the structure of the DNA, but can be also approached by using molecular dynamic simulation. If you change completely systems, so if you want to look how type of information we can extract by using UV spectroscopy in the case of peptide and the protein, we can see that if you change the excitation wavelength, we can study more in detail the secondary or tertiary structure of protein. And in particular, is it possible to isolate in the spectra different molecular portion, mainly associated to the amide group and aromatic amino acid present in the protein and peptide. Here we can see a typical example of the spectra that we can collect by using excitation wavelength at 226 nanometer that is in close resonance with the electronic transition of aromatic amino acid. And this is the spectrum of the beta-lactoglobulin protein in relative dilute solution, where the dominant band are associated to the tryptophan and tyrosine amino acid present in the sample. But in particular, if you look to this, the intensity of this band associated to the tryptophan, the intensity of this Raman signal are very sensitive to the surrounding chemical environment of the tryptophan, and in particular the Raman cross-section. So the intensity of this band is able to detect the increase in the water exposure of the tryptophan that occur, for example, when the protein is unfolded. This is the case of this graph where we follow the intensity of the tryptophan band as a function of temperature. And this graph mark the increment of the exposition of the tryptophan, so the more hydrophobic part of the proteins, related to the folding of the protein. In particular, here we can detect this increment of the curve, detect the presence, the occurrence of the melting transition of the protein. But we can see that using this technique, we are sensitive also to other events that occur at lower temperatures, but to the melting of the protein, that consists in conformational change of the protein that can be detected specifically by using this technique. Here we can see another example where the modification in the protein aggregation state, in particular the formation of this fibril, this is the case of the lysozyme, produce a strong change in the spectrum of the protein, in particular in the intensity of the signal associated to the tryptophan amino acid, and this spectral change can be associated to the modification in the tertiary structure of the protein that can be efficiently monitored by using UV spectroscopy. In a similar way, we can follow the UV spectroscopy can detect different protein aggregation paths. In this case, the case of this study that concern the preparation of this hydrogel, protein hydrogel, by using beta-lactoglobulin. We prepare this concentrated solution of the protein that at different pH as a function of temperature show different behavior. In particular, in the presence of acid pH, we observe the formation of this amyloid-like aggregate at the formation of this hydrogel of protein, and this is can be in a very can be detected marked by this decrement of this signal associated to the tryptophan. From the other side, if you use the basic pH, we observe another type of transition for the protein from the folded to the molten globule state that is marked by the different behavior, in particular by the increment of the intensity of these signals. So the intensity of these signals are representative also of different processes occurring in the protein. Here there is another example of the effect of pH on the structure of this protein, concanavalin ACE structure. Also, in this case, we follow the evolution of the spectra as a function of temperature, and we can identify in the spectra specific spectral markers that are sensitive, for example, to this irreversible aggregation of the protein, and in particular to the formation of this beta aggregate that is modulated by different pH. The analysis of these spectra can be associated also to the chemometric method that help to obtain more detailed insight from the analysis of the spectra, and this is a work in collaboration with a group of Professor Sato, who is in the audience today. And this is another possible application in the case of the fibrinodular structural change. Here we are interested to study the structural change of the protein at the surface of carbon nanoparticle. In this case, we can detect specific spectral marker that can mark the unfolding and aggregation event occurring in the protein. And in particular, in this study, we have found that the presence of this nanoparticle, carbon nanoparticle, in solution tend to mitigate the effect of the temperature. And also in this case, we are collaborating with a group in Barcelona for the development of machine learning methods for the analysis of these spectra. We are developing specific protocol that can be applied to the analysis of these UV spectra. In particular, in the case of smaller molecules, so for example, peptide or short polypeptide, we can associate the experiment also with DFT protocol for simulation. In particular, we had a collaboration with a group of theoretical chemists in Scuola Normale of Pisa. They develop code, in particular quantum mechanical model to reproduce experimental spectra also Raman and Resonance Raman spectra of molecule in solvated state. This is a very important point because usually in quantum mechanical models treat the solvent as a continuous it's very important to take account also the specific hydrogen bonds of the solvent at specific surface of the, in this case of amide or small dipeptide. It's very important to take account of this effect because here we have demonstrated, for example, that are specific effect that we detect in UV resonance Raman spectroscopy. For example, the selective enhancement of this band that is called amide 2 band. The spectra of protein or peptide are characterized by the presence of these bands that are associated to the amide bonds present in the sample. In the visible Raman spectra, the amide 2 bands. These bands are very interesting because are strongly related to the secondary structure of peptide and proteins. So are usually used also in a qualitative way in order to establish also in very quantitative way the fraction of different type of secondary structure present in the sample. But in the case of visible Raman spectra, the amide 2 mode is completely absent. In the UV resonance Raman spectra, we start to see the rising of this amide 2 band that become dominant, especially at shortest excitation wavelength, and in particular we can observe this selective enhancement of this band that tend to increase more with respect to the other signal when we go toward the resonance of the amide group. But we observe this specific effect in dipeptide and tripeptide only when these peptides are in hydrated state. So this means that this selective enhancement is related to the solvation effect, and this solvation effect or this experimental finding can be reproduced only if your quantum mechanical model for the simulation we take account of the so-called explicit water molecules. So we take account of the specific hydrogen bond that water molecule can make with specific site of this dipeptide in this case. So by introducing this explicit water molecule, we are able to reproduce the experimental effect. So we demonstrated that this effect is strongly related to the hydrogen, the amide spectra fingerprint are in general hydrogen bonding mediated. And this is very interesting also for the possibility of the development of new code for the simulation of this small peptide. This type of information are very important when, for example, we try to interpret the spectra of small peptide. This is another true example of a collaboration that we have with the group of Silvia Marchesan, Professor Silvia Marchesan in the University of Trieste, where we use UV spectroscopy in combination with other technique to detect the self-assembly of small peptide. Here I report 2 different examples of small peptide that tend to assemble in a specific structure and form gel or crystal phase or other aromatic small molecule that tend to form this cage. And in this case, the formation of this structure is very important. These stacking interaction that are the interaction of the that are developed when these aromatic part are close all together, and this is in this case, the UVRR technique is able to enhance in the Raman spectra the signal selectively related to these aromatic parts. So we can extract information on these specific interaction that are fundamental for the definition of this structure by using this technique. Finally, I would like to show just some preliminary results of work in collaboration with Alessandro Marcello and Denis Reinovich of HGB. We hope that this proof of concept study, this is the first step of the work that we plan, part of the work that we plan on the PRP project. And so the idea to use UVRR spectroscopy for the selective detection of pathogens. In this case, we work on this model vesicular stomatid virus. For the preparation protocol, it's better if you ask to Dennis, that is not here today, but the important thing from my side of the spectroscopic point of view is the fact that if you change the excitation wavelength, here we can see that in the spectra of the pathogens of the virus, we can identify different biological markers that are related to the different part that compose the virus, in particular RNA, lipids, and protein. In the spectra that we collect using an excitation wavelength at 266, the spectra are dominated mainly by the signal associated to the adenine, guanine, and thymine nucleobases of the RNA component. At the other side, the excitation wavelength at 213 nanometer is able to enhance the signal mainly related to the lipids and the protein part that compose the system. And the important thing is that we collect this spectra in the sample in the solvent and PBS buffer in relative real conditions, so just after the purification put in the sample holder, and we measure the spectra without any particular manipulation. And in fact, we use this approach also, for example, to investigate in real time the deactivation of this virus related under the irradiation at 213 nanometer. The idea in this case was to investigate the possibility to build an effective tool for virus deactivation that use UV light, but at energy that are not so dangerous for the human skin as the UV lamp that are usually used at the moment for this type of the deactivation. And here we can see that if you collect the spectra as a function of time, we can identify specific spectral marker that change as a function of the time of radiation, and we can use this spectral intensity, in particular of this spectral marker, as a sort of quantitative evaluation of the deactivation of the virus that after 1 hour is completely deactivated. The next step of this study is, for example, to use SARS-CoV active virus. At the moment, the problem is to find the way to transport sample that can be treated in biosafety level 2 or 3 on the beamline that are environment not studied for level 2 or level 3. So, but we are working in this sense to develop also protocol for the safety measurement for this type of experiment. Okay, so in conclusion, this is our team of people, scientists who work on the beamline and collaboration, visiting and other people. And thank you for your attention.